Nothing fills a room like the promise of smut. Thank you all for having me here. I'm very happy to be here at McMaster. It's been a lot of fun meeting everybody, and it's inspiring to be in a place that is genuinely interdisciplinary. So it turns out that I'm a little bit of a fraud here. My research actually isn't about love or sex or brain evolution. And my love life is an utter disaster. So really, think of me not so much as an expert, but as an ambassador from the neuroscience nation trying to explain some work that's mostly actually been done in the laboratories of other people. So our brains are a jumble, a kludge, a workaround, and a pastiche. They weren't designed all at once by a genius inventor on a blank sheet of paper. And tonight I hope to convince you that our human experience of love and sex ultimately derives from the properties of our inefficient and bizarre brains, which are a weird agglomeration of ad hoc solutions that have accumulated through millions of years of evolutionary history. Now, one can fashion explanations of human amorous and sexual behaviors using the methods of cultural anthropology and social history, and these are entirely valid and useful endeavors. And certainly ideas and practices involving human sexuality are deeply influenced by culture. However, what I'm seeking here is a different type of understanding, and it's one that's less nuanced but perhaps more fundamental, a cross-cultural biological explanation. And what I'd like to do is to start by considering two very mysterious observations about human behavior. And the first one, I'd like to take you to Oxford in the 1970s, the laboratory of the vision scientist Larry Weisskranz. And Larry Weisskranz did an experiment which on the face of it seemed utterly insane. He recruited blind people who had lost their sight as a result of a blow to the back of the head. So their eyes were intact, but they were entirely blind as a result of damage to a region of the brain called the visual cortex. And he put a letter in the hands of these subjects, and in front there was a letter slot, and it was either oriented vertically or horizontally. And he said, what I'd like you to do is put the letter in the slot. And the subject said, are you kidding? What a silly idea. I'm utterly blind. I would be doing it totally randomly. And Weisskranz said, well, look, just go with your gut and put the letter in the slot, and we'll see what happens. And so he did this with a whole bunch of these blind folks. And it turns out that actually most of them could orient the letter to the slot, not perfectly, but at levels that were much, much higher than chance. Do we have to get all woo-woo about this? Right? This is a science seminar series, right? We're not supposed to get all woo-woo. All right, let's put that one aside for a while. We'll come back to it. Some of you might know the answer. So what does that have to do with sex and love? Well, we'll get there. What is your cat thinking when she watches you have sex? Well, I'll tell you. Even if you're as sexually conventional as they come in this culture, let's say you're in a committed heterosexual relationship and you're not into all that kinky stuff. You're not dressed up in a Dick Cheney rubber mask with clamps on your nipples. What's the appropriate Canadian equivalent of a Dick Cheney rubber mask? I should have a more local, what would that be? All right, okay, thank you. With clamps on your nipples and Wagner's Ring Cycle playing in the background, you haven't inserted a Bluetooth-enabled electrical shock probe in your anus that's connected to the internet to be triggered by wild fluctuations in the Hang Seng Stock Index. Rather, you're with your partner in private, in your bedroom, hugging, kissing, petting, licking, having vaginal intercourse, the usual, total conventional scene. Your cat still thinks that you're a goddamn freak. And she's right. She's revolted by the fact that you mate during the female's non-fertile time. That's just disgusting. And she thinks that you're a moron for sticking with one sexual partner given a single ovulatory cycle. This makes no sense at all. And this privacy thing, that's just weird. Everyone knows you're supposed to have sex in public where the social group can watch and join in if necessary. And finally, if a baby results from this lovemaking, she'll wonder what the male is doing, hanging around all the time, helping out, providing resources, the wimp. And don't get your cat started on that child. 5 years old and it still can't take care of itself? What a loser. So as your cat knows, humans are off on the fringe here. And most other mammals, the female advertises her fertility with clear signals, unique sexual gestures and calls, odors and swellings and so on. Males and females do not typically approach each other for sex outside of these fertile times. By contrast, human females have concealed ovulation. There are no clear displays of a woman's ovulatory cycle. And one consequence of this is that most human sex, even most human penis-vagina intercourse, is recreational. It's not timed to the ovulatory phase. It even continues in situations where conception is completely impossible, such as during pregnancy or after menopause. Another way in which humans are aberrant has to do with our choice of sex partners. Over 90% of mammalian species are highly sexually promiscuous. So both males and females have multiple sexual partners on the same day. Humans tend to be monogamous, or at least serially monogamous. And what's crucial here when we speak about the relative monogamy of humans is that most women have a single sexual partner in a given ovulatory cycle. And as a consequence, unlike other mammals, knowledge of human paternity is very accurate. So when genetic population studies are performed, over 90% of children are found to be the offspring of the mother's long-term partner or husband. And it doesn't matter if the study is done in Toronto or Beijing or a tiny village in Papua New Guinea, you get the same answer. The social attribution of human paternity is extremely accurate. So finally, and perhaps most importantly, in almost all mammalian species, the male and the female do not form a lasting pair bond after mating, and so the male has no role in rearing the offspring. In fact, in many cases, the male will just leave the social group entirely, and if he stays, he's unlikely to recognize his own progeny. In humans, lasting pair bonds are common, and the man typically contributes to the well-being of his offspring, even if it's not through direct care. So, of course, in very recent years, changes in social conventions and changes in technology have allowed single motherhood to be a very viable human endeavor. But actually, if you look worldwide, it's still very rare. And on an evolutionary timescale, it has only appeared at the very, very last moment. So, what are the answers? Why do we have this phenomenon of these blind people orienting the letter, which Weisskranz called blind sight? And why do we have such a weird mating system? And these are the reasons. They have to do with evolutionary constraints on brain design, and I've summarized these as follows. The brain is built like an ice cream cone, and neurons as information processors suck. And I gave this lecture in Holland, and I said neurons as information processors suck. And they all looked at me blankly, even though they spoke very good English. And I said, well, suck is a North American term that means doesn't work very well. And they said, oh, yeah, okay. You get it. So what do I mean by the brain is built like an ice cream cone? So when you are an engineer designing a car, you might have a design that comes out in a certain year, and then in subsequent years you might refine it very minimally, but every once in a while you'll entirely refresh the line and redesign the thing from the ground up. And that's not how evolution works. Evolution is agglomerative. Generally speaking, not uniquely, absolutely, but generally speaking, brain evolution has proceeded by adding systems onto existing ones. It's kind of like putting scoops on an ice cream cone, right? So if you have an ice cream cone with one scoop and you want to put a second scoop on it, you don't remove the first scoop and redesign it and put it on and then add the second scoop. You just put the second scoop on the first scoop. And then you put the third scoop on top of the second scoop if you're really a pig like me. And so if we look how brain evolution has proceeded, generally speaking, the parts of our brain which are evolutionarily the oldest, which are literally the lowest in our brain, the closest to our spinal cord. And so these are regions like the brainstem and the cerebellum and the midbrain. Their fundamental architecture and their fundamental electrical and neurochemical function is only slightly different than what you would find in a lizard. And if you've got a lizard brain and you want to build a mouse from your lizard, well, you don't completely redesign those lowest parts of the brain. You add on, you modify the midbrain a little bit, and you add on a limbic system, which is an emotional and regulatory core system of the brain. And if you want to take a mouse brain and you want to make a primate brain out of that, well, you expand the topmost, outermost rind, called the neocortex, tremendously. And if you want to go from a chimpanzee to a human, you grow that cortex even more, particularly in the front half of the brain. But you never get to do a total redesign, and you are fundamentally constrained by choices that were made very, very early. And so our best estimate of when nervous systems began to evolve is on the order of 600 million years ago, when there are creatures in the fossil record that look like modern-day cnidarians. And this is a group of animals that includes radially symmetric animals with stinging cells like jellyfish and corals. And they have neurons. And modern-day cnidarian neurons are basically similar in their plan to modern-day human neurons with some fairly subtle alterations. So what does this mean in terms of brain design? Well, it means that many times in the brain you actually have multiple systems in parallel. So it turns out that in your brain you don't just have one visual system, you have two, and some would argue actually even have more than two. But there are at least two main ones. There's the evolutionarily modern one, so you have information that flows from your eyes, and it flows through a relay station, your thalamus, to your visual cortex. And this is the beginning of a processing pathway that gives rise to your conscious visual perception of objects. And this is the area that is decimated in Weizkranz's patients, and it is why they cannot have any conscious visual perception. However, there is a second stream that goes to an evolutionarily ancient visual center called the optic tectum in the midbrain. And this visual system, which we share with animals down to lizards, for example, is very good at things like helping to orient your eyes and head to motion in the periphery of your visual field. It's very good if there's an object that's expanding symmetrically in your visual field, so it looks like it's about to impact you in the nose, giving you your reflexive ducking reflex. And, but information to this evolutionarily ancient visual system does not have access to your conscious mind. So in Weizkranz's patients, they are able to take that visual information that's flowing from the eyes to the evolutionarily ancient visual system. It can guide their decisions subconsciously, even though they are not consciously aware of it, and cause them to orient that letter appropriately at levels far beyond chance. All right, now, okay, that solves that problem. What about love and sex, though? So neurons as information processors suck. What do I mean? Well, we know that neurons send electrical signals, and we're used to thinking of electrical signals in electronic devices moving through insulated copper wires, for example, that moving at speeds close to the speed of light. And it turns out that electrical information in neurons moves much slower. So the very fastest electrical information in neurons moves at about 200 meters per second, and the very slowest moves at about 0.1 meters per second. And the fundamental act of information transfer in a neuron is when an action potential, a large positive deflection in the voltage gradient across the membrane of a neuron, enters the information sending end of the neuron called the axon terminal that causes it to release neurotransmitter. Neurotransmitter is a chemical that there are many of them. They diffuse across a narrow fluid-filled gap called the synaptic cleft, and they bind neurotransmitter receptors on the next neuron. I think many of you have heard of this process. And those neurotransmitter receptors can then have electrical consequences that propagate signals further in a chain of neurons, giving rise to neural computation. So the release of neurotransmitter is a fundamental act in the processing life of a neuron, and you would imagine that it might be extremely reliable, but it turns out that it isn't. It's highly probabilistic. At most synapses in your brain, when an action potential invades a terminal, it only releases neurotransmitter about 1/5 of the time. Your brain is rolling dice with information, with all apologies to Einstein. Neurons have a severely limited signaling range. They can go from 0 spikes per second up to a very maximum of about 1,500, 2,000 spikes per second. And they leak signals to their neighbors constantly. So to give you an idea of the speed of neural processing, what I always tell my medical students at Hopkins is that if we imagine that there's a giant and she has her toe hanging off the tip of South Africa and she has her head in Baltimore, and a great white shark bites her on the toe on Monday, she won't feel it until Wednesday, and she won't jerk her toe until Saturday. So neurons as information processors suck. And so then you've got this issue, right? How do we build clever us out of such crummy parts, right? We're great. You know, we're terrific at cognition, right? We can look at an image of a Rottweiler from the front, and we can look at an image of a teacup poodle from the back, and we instantly know that these both belong to the category dog. Now that turns out to be a very difficult thing for a computer to do, but a very simple thing for a human to do. We're pretty good at using our brains in the world. So how do we get clever us from these lousy parts? And I think you know the answer. And the answer is, in order to do that, you've got to have an extremely large number of these lousy parts of these neurons. So in the human brain, we're talking on the order of 200 billion neurons. And you have to have them massively interconnected. So each neuron, on average, is receiving between 5,000 and 10,000 contacts from other neurons in the brain. And so when you do the math, you're, you know, kind of all on the order of 500 trillion to a quadrillion synaptic connections. All right, well, that is a big, fat head. Right? So there's a couple of really big problems that come up here, right? So in order to build a clever brain, you can only add stuff on. You can't take stuff away. So like, imagine Someone said to you, all right, I've got a great job for you. You're going to build the world's greatest speedboat. And you go, oh, great, I'd love that job. Sign up. Well, okay, actually, we didn't tell you. We're going to give you a dinghy, and the way you build the world's greatest speedboat is you can only add things to your dinghy, but you can't take anything away. And that's basically what's happened in brain evolution. You've just glommed stuff on. So the fact that evolution of the brain proceeds mostly through agglomeration, and the fact that these neurons are lousy processors means that we need a huge, fat, 1,200 cc adult size human brain to be clever. And so there's one problem with this, right? So we got these big fat brains and these narrow vaginas, right? And so you women in the audience who've given birth know that this is a little problematic. And it turns out that death in childbirth is almost exclusively a human phenomenon. You don't find it in almost any other mammal with the exception of the hyena. The hyena female has a pseudophallus that it gives birth through, which is kind of a weird case. But leaving the hyena aside, death in childbirth is exclusively a human phenomenon. And the volume of the brain at birth of a human infant is about 400 cubic centimeters. And that's really pushing the limit. That's about as, there's problems even there, right? You got this 400 cc brain. It's got to become a 1,200 cc adult brain. And here's an issue. So you've got all these neurons. They've got all this crazy wiring that you need to build clever us with these shitty parts. And it turns out that that wiring, of course, isn't random, right? There is a wiring diagram for the brain. So then, like, when you get into all these trillions and quadrillions of connections, how do you specify that wiring diagram? Is there even enough information in the DNA and the genome to specify that wiring diagram? And the answer is that it turns out that the wiring diagram of the brain is only roughly specified genetically, and at the very finest level, it turns out to be guided by experience. And that experience actually starts in utero, and it proceeds in early life, and it proceeds at a very fast pace, furious pace, until about age 5. And it proceeds at a much slower pace, but isn't done until about age 20. So the consequence of these constraints is that we humans have absurdly, crazily long childhoods. So why then did humans end up with our grossly atypical mating system? Why did we end up with love? And the most compelling theory is that the human mating system has been driven by the fact that humans have the longest, most helpless childhood of any mammal. So while an orangutan or a gray whale mother can raise her offspring successfully without paternal involvement, human single mothers in traditional societies are at great disadvantage because their offspring remain helpless for so very long, as your cat knows. Human children with their slowly maturing brains are the reason for our atypical mating system with concealed ovulation, mostly recreational sex, reliable paternity, monogamy within an ovarian cycle, and paternal contribution to child rearing. Or to put it another way, if human neurons weren't such lousy processors, and if brain evolution allowed for redesign from the ground up, rather than proceeding mostly by agglomeration, then we wouldn't need such big fat brains and such long childhoods, and heterosexual marriage would not exist as a dominant cross-cultural institution. So, to this point, I've concentrated on the generic sexual norm of the dominant human culture, the kind that established religions tend to like: monogamous, heterosexual, procreative. And we tend to imagine that other mammals with their limited access to the internet are less adept at expressing their kinks. And in this vein, it's worth mentioning that there are many common but less broadly sanctioned expressions of human sexuality that are also seen in other species. So masturbation is a common practice among a number of mammalian species, including horses, monkeys, dolphins, goats, and elephants. Now, I want you to use your imaginative faculties here. I don't want you to just be passive recipients of this information. I'd like you to create in your mind's eye all these images. Both males and females indulge in this pleasure, and like humans, they've been quite inventive in its pursuit. Males of a number of species engage in autofellatio, and there are also a few notable reports of autocunnilingus among primates. Female orangutans have even employed crude dildos fashioned from tree bark and sticks. But my award for the most creative animal masturbation has to go to the male bottlenose dolphin, which at least in captivity has been observed to wrap a live wriggling eel around its penis. Don't try this at home. So Bruce Vagmihi of the University of Wisconsin states that homosexual behavior has been well documented in over 500 species and is likely to be present in many more. And this includes both male and female homosexual behavior, although the former has been more widely observed. Mammalian homosexuality is expressed in all the ways you might imagine and in some that probably hadn't occurred to you. So male-male and female-female oral sex has been documented in a number of species, including hyenas and bonobos, which are pygmy chimpanzees seen here, as has female genital-to-genital rubbing. So these are actually two female bonobos involved in genital-to-genital rubbing. Male-male anal sex has been seen in sheep and giraffes and bison, and male bottlenose dolphin pairs will insert their penises in each other's bottlenose dolphin pairs will penetrate each other's genital slits, and male Amazon River dolphins will even insert their penises in each other's blowholes in the only known example of nasal sex. So if you got nothing else out by coming to this lecture tonight, you can take that anecdote home and impress your friends, right? Did you hear about the Amazon River dolphins? So most homosexual behavior seen in other mammals would be more accurately described as bisexuality. In many species, heterosexual content, as we mentioned before, only occurs during the mating season, but homosexual behavior is common at other times. In some, like the bonobo shown here, it seems as if homosexual behavior, in addition to providing sexual pleasure, also fulfills a social role to diffuse tension and promote social bonding at the expense of aggression. And bonobos are, as you may have heard, they're an unusually peaceful, matriarchal primate group, unusually so. So to recap this portion, we've got mammals involved in gay and straight sex of the vaginal, oral, anal, and even nasal varieties, and masturbation, sometimes with the aid of objects, and I won't get into the details of it, but cross-species sex and animal necrophilia have also been observed. So in the end, it's a bit counterintuitive. It's not really the kinky or the forbidden stuff that makes us humans sexually unique. Those things are well represented in our mammalian kin. Rather, it's the most conventional and socially sanctioned mating behavior that is totally aberrant in comparison to other mammals. So I'd like to turn to the topic of love, or lerv, as we like to say. So we know that intense romantic love is not just a modern idea. It's expressed in some of the oldest surviving writings from places like China, Egypt, Greece, and Sumeria. But is it truly a cross-cultural universal, or is it just an aspect of a handful of cultures? And to address this question, Helen Fisher, who's an anthropologist at Rutgers, surveyed data collected by cultural anthropologists from 166 different societies. And she found evidence for the idea of romantic love in 147 of these. And in the remainder, there was no clear indication that romantic love did not exist, and rather these were just cases where the anthropologists hadn't asked about the topic. Now, parenthetically, just because the idea of romantic love exists doesn't mean that it is actually allowed by the society to be put into practice as the norm, right? So the description of the mental and physiological aspects of intense romantic love are surprisingly similar across cultures. And so we're talking about intense, giddy pleasure, suppression of appetite, distortions of judgment about the beloved, accentuating the good traits and diminishing the bad ones, and distortions of judgment about the world. We've been so discreet, no one knows. As well as obsession and, of course, sexual desire. And there's a crucial feedback loop in new love. We not only see all the wonderful things in our beloved, but when we look into their eyes, we see those same positive feelings mirrored back. Fundamentally, when we're in love, we like ourselves better as well. So then the question becomes, you've got these description of mental and physiological aspects of intense romantic love that are surprisingly similar cross-culturally. So what are the neural correlates of those? And so Lucy Brown from Albert Einstein in New York City did a study where she recruited people who were in very new love relationships within the first 8 months or so, people who describe themselves as intensely, madly, passionately in love. And she had them view faces, view pictures of their lover's face while they're inside a brain scanner. And as a control, she had them view pictures of a platonic friend who was equally familiar. And then basically what she did is she subtracted the pattern of activation that you got with the platonic friend from the activation that you would see with the lover's face to try to reveal those patterns of activation, those patterns that were unique to the lover's face. And so when she did this, she found a number of areas that were activated and a number of areas that were deactivated. And so here's showing one of these areas that's activated. It's called the ventral tegmental area, or VTA, a region in the midbrain. Now, those of you who are older may know a rock and roll song called "Love is the Drug" by Roxy Music. And it turns out that, well, they were right. It turns out that this ventral tegmental area and its primary target region, the nucleus accumbens, these are the same areas of the brain that are activated by drugs like heroin and amphetamine and alcohol and nicotine and cannabis. They comprise a central portion of what's known as the medial forebrain pleasure circuit. Now I'm showing you a different view of the brain, and this is a rat brain, not a human brain, but it's a very similar structure. So here's the ventral tegmental area. It has cell bodies shown here in white, and these cells project their information sending ends, their axons, to a number of brain regions, including the nucleus accumbens, which we discussed, and the dorsal stratum, and the prefrontal cortex. The prefrontal cortex is a logic and cognition or reasoning center, the so-called executive control center in the brain. The amygdala is involved in emotional learning, particularly for fear. The hippocampus has a special role in the storage of memory for facts and events. And these neurons use the neurotransmitter dopamine, and they're strongly activated by a wide range of pleasurable stimuli, including not just vices, but also virtues like donating to charity, or exercise, or even paying your taxes. So I guess it's not that surprising that when you're looking at the new love's face, this pleasure circuit lights up. There's also another area that lights up in the cerebellum. The cerebellum is this baseball-sized thing that hangs off the back of your brain that's involved in motor coordination and motor learning. So if you sustain damage to your cerebellum, you're not paralyzed, but you become very clumsy. Now, why the lover's face should activate this region in the cerebellum is not clear. But what's even more interesting is to look at what is deactivated when you look at the lover's face. Well, the prefrontal cortex, that's an executive reasoning area. The temporal poles and the parietotemporal junction, these are crucial areas for social cognition. So if you look at the mental and physiological aspects of intense romantic love, right? Distortions of judgment about the beloved, yeah, we're right there, okay? Giddy pleasure, we're right there. It actually lines up with the patterns of brain activation and deactivation very, very effectively. Now, it turns out that this pattern of activation is the same for men and women. It's the same depending if you're straight or if you're gay. It is the same depending on whether you do the study in Beijing or in New York. It's now been done in both places. So you might ask, all right, well, these are new, madly, passionately in love lovers looking. What happens when you do this with old married couples who've been together a long time? And so Lucy Brown got her cohort and she interviewed these people and she said, is your love for your partner as intense and fresh now as it was when you first met? And 19 out of 20 people said, no, it isn't. It's not that intense anymore. It's mellowed into a different thing. And 1 out of 20 people said, yes, it is just as good. And you might think, all right, well, these people have watched too much daytime TV, right? You know, they're just high on Oprah and, you know, they want to appear a certain way, so they're saying this, but, you know, all right. So Lucy Brown put all these people in her scanner, right? So the 19 out of 20 people looking at the pictures of their longtime lovers, you don't see any of this stuff. You don't see the strong activation of the pleasure circuit or the deep nuclei. You don't see deactivation of the prefrontal cortex or of these social cognition areas. You are seeing your long-term partner very, very, very clearly. So what about that 20th person? Well, I'll be damned if these people who self-reported this, who I would just have assumed were just full of shit. Sure enough, they have the same pleasure circuit activation that you would see in new lovers. And they don't particularly have, however, the deactivation of these reasoning, executive control, and social cognition areas. So maybe they found the best of both worlds. Now, you might imagine, well, you got new lovers, and they're looking at pictures of their beloved. Maybe they're just feeling horny, right? In other words, maybe what you're measuring here is not love per se, it's just sexual arousal. They're looking at a picture of their new lover's face, they're thinking about what they're going to do later. Okay. So what happens if you contrast this pattern of activation with brain scans of people who are just watching porn? And of course, you know, you've got to do this with matching people to the porn that's to their liking, if they're gay or straight or into this or that or whatever, right? You know, so you get people watching porn that they rate subjectively as arousing, and then you put them in the fMRI and you see the pattern of brain activation. And it turns out that these regions are activated watching porn, but the deactivation of these cognition regions doesn't happen watching porn. In a way, that's not that surprising, right? Your social judgment isn't utterly tossed out the window. We knew this already. We know that feeling in love and feeling horny are two different things, right? It's nice to have it confirmed. All right, so I'd like to turn to the topic of orgasm. Now, I think this is an obvious thing, but I'm going to say it anyway. Orgasm occurs in the brain, not the crotch, and it can happen in sleep. It can happen with thought alone, with no peripheral stimulation. It can happen in cases of people with complete spinal cord transection. It can happen with stimulation of non-genital regions. And in some people, it can occur as a result of seizures. So it turns out that if you talk to neurologists, seizures are often triggered by repetitive actions. And one of the repetitive actions that's a particularly good seizure trigger is toothbrushing. Right? So a lot of people brush their teeth, they get into that rhythm, and it triggers a seizure. And I'd like to show you the EEG of a woman in Taiwan. She started brushing her teeth at this moment. This is her EEG. Everything's going along fine. Her seizure started, and at that very moment, she had an orgasm. And it turns out this wasn't just a one-time thing. Every time she brushes her teeth, she has an orgasm. And you might think that this woman would have the world's best oral hygiene. But it actually turns out, when you think about it, this isn't the case at all, right? I mean, you know, you don't want to have an orgasm while you're brushing your teeth. You just want to get out the door and get going. She actually finds this very disempowering and very distressing. And it's you know, as much as we make light of it, it's actually a big problem. And it turns out that in this particular case, that the orgasm that is evoked by the seizure, that the seizure involves broad regions of the brain, and this woman has a conventional orgasm. Now, what do I mean by conventional? Well, it turns out that some people can have seizures that give rise to orgasms that are not pleasurable. You might think, well, how can an orgasm be not pleasurable? Aren't orgasms intrinsically pleasurable? Isn't that, well, so, you know, when we experience orgasm, typically it's a unified percept, right? But it turns out that it's a theme in sensory systems, and orgasm is one example of this, where it turns out that there are separable sensory discriminative sensations that involve one particular region of the brain. So, for example, the somatosensory cortex gives rise to the sensory discriminative aspect of orgasm. But the emotional and affective part of orgasm actually depends on a different structure. It depends upon the anterior cingulate cortex here and another adjacent region called the insula, more towards the midline of the brain. And so it turns out that if you have a seizure that involves the somatosensory cortex and activates the genital regions, you can have an orgasm that is more like whoops than like what you would normally experience. The same thing can happen in painful situations. So there are people who have damage to these midline portions of the brain, and they have pain that has no emotional component to it. So you take them in the lab, the standard pain test, it's called the cold pressor test. You put their hand on a bucket of ice water, it hurts like hell. I volunteered for this test. It's really unpleasant. And most people will go, oh man, that really hurts, and they'll have an emotional reaction. The pain will have an emotional resonance. And someone with damage to this medial system will say, yep, that hurts. Boy, does that hurt. Yeah, it hurts a lot. But there will be no emotion in their voice, and they will not be experiencing it as emotionally resonant in much the same way as this non-pleasurable orgasm didn't engage the emotional component. Now, it turns out that there are people who have damage to somatosensory cortex in which they have lost the sensory discriminative aspect of pain, but they keep the affective emotional component. Oh my God, it hurts like hell, it's awful. Where on your body does it hurt? I have no idea. Right? So our brains are taking our stream of sensory information, they are blending it with emotion, and they are presenting it to us as a unified percept. And it's only in these unusual cases that the curtain is dropped and we see the separate subcomponents. So, what's happening in orgasm in the brain? So, orgasm physiology is remarkably similar in men and women. In both sexes, orgasm involves an increase in the blood pressure, rising heart rate, involuntary muscle contractions, including the muscles of the rectum and the urethra, and of course an intensely pleasurable sensation, unless you have this unpleasant brain damage. So women's orgasms, as measured by a rectal probe, are on the average slightly longer than men's, about 25 seconds, as compared with 15 seconds. Now, what brain regions are activated during orgasm? So now, I want you to actually imagine what this experiment looks like, right? And this is how it's done in Geert Holstege's lab at the University of Groningen Medical Center in the Netherlands. Now, of course, it's hard to get human subjects review board approval for this kind of work, and so most of it happens in the Netherlands, it turns out. So the subject's head is fixed tightly in place with an adhesive band, and it's inside this giant metal donut that just comprises the brain scanner. Imagine that your subject, she's hooked up to an IV line with a needle in her arm to deliver the pulse of radioactive water tracer that's required for this particular form of brain scanning called PET imaging, or positron emission tomography. And a pressure transducing probe is inserted to measure the rectal contractions that accompany orgasm. And she's instructed to close her eyes and to keep as still as possible to minimize brain activity that's unrelated to orgasm. Now, can you imagine a less sexy situation than this? Right, so then the woman's partner begins manual stimulation of her clitoris, and when it appears as she's approaching orgasm, she gives a sign. The tracer is injected through the IV line in her arm, and they say, okay, hope it happens in the next 60 seconds while the tracer's in your brain, because after that, it's too late. No pressure. So it's kind of amazing that anyone can achieve orgasm under these conditions. It actually turns out that women are much better in this situation than men, and most of the male subjects, before the invention of erection-promoting drugs like the phosphodiesterase type 5 inhibitors like Viagra and Cialis, were unable to complete this kind of experiment. All right, so what do we see? All right, first of all, the rectal probe, right? I can't tell you, as a fan of accounts of people abducted by aliens, I can't tell you how much pleasure it gives me just to say the words rectal probe. So, the rectal probe doesn't lie. And if you're concerned, if you men are concerned that your female partner is faking it, so in this experiment, this is the rectal probe from a woman who is having a real orgasm, and you can see these contractions, and they go on for 25, 30 seconds or so. And the control here I thought was really brilliant. The black line, they asked the woman to fake it. And the woman faked it. You can fake it, but you can't fake the rectal probe. So the rectal probe is very useful in these experiments because it allows you, if you want to align the brain scanning images and be confident that you are getting a brain scan that actually includes the time of orgasm, then obviously you want to have, don't want to just trust the person's subjective report, you want to have an independent measure of orgasm. And so all humor aside, the rectal probe actually serves a good function here. So what do we see? By God, we've seen this list of brain regions before, haven't we, right? So we see activation of the pleasure circuit by orgasm, the VTA and the nucleus accumbens. We see our friend, the deep cerebellar nuclei activated here. We see deactivation of our executive and our social cognition regions. And at the moment of orgasm, you're not thinking that clearly. And interestingly, in males, but not in females, you see activation of a brainstem region called the periaqueductal gray that is heavily endowed with endorphin and enkephalin-containing neurons. And this may or may not have something crucial to do with male sexual experience. Now, in terms of the deep cerebellar nuclei and orgasm, this is a little bit easier to understand than the New Lover's Face study. Well, maybe not. But the cerebellum basically exists. It is a structure, it's an engine that compares plans for motor action with feedback about how that action is processing. So if I'm not paying attention to what I'm doing and I'm walking along and at this moment, whoops, I unexpectedly bump into something, right? You would see activation of my cerebellar deep nuclei, right? Because my plan for motor action and the feedback, it's how it's progressing, there was a mismatch detected. Well, in a way, orgasm is the ultimate sensory motor mismatch, right? So you're doing some motions and you're getting some sensory feedback. You're doing some motions, you're getting some sensory feedback, and then things kind of get out of control, right? And it goes off into a region where the sensory feedback isn't entirely predicted by your motions. So in that sense, an orgasm is a motor error, like another motor error, and perhaps it's not surprising that that you see the cerebellar region activated in orgasm. So it turns out that with the exception of the periaqueductal gray, these patterns of brain activation are remarkably similar in males and females. And this validates a very interesting previous study in which 24 male and 24 female subjects were asked to write description of their orgasms, and then somebody edited them to redact the gender-specific information about certain body parts or anything that would be particular. So what you were left with was a description that you couldn't uniquely identify. And then these descriptions were given to a panel of gynecologists and urologists. And they tried psychologists, medical students. Nobody could distinguish the male and the female orgasm descriptions from each other, which perhaps, given the similarity in the patterns of brain activation, is not that unusual. So, step back a bit. What can we conclude? So, those things we hold as most central to our human experience, like romantic love and long-term pair bonding, are not the ultimate design features of an impeccably engineered brain, and furthermore, they don't happen despite the quirky, evolved structures of our brains. Rather, we have these features precisely because of our evolutionary history. Thank you very much for your attention.